Okay, so welcome back, all of you, and to the final session, the keynotes from Professor Andrea Dunaif from Icahn. Oh no, it's better you present yourself, but you are at Icahn School of Medicine, Mount Sinai. She is really the PCOS lady in the world, I must say. You have been working with PCOS, done fabulous work on mechanism of try to understand the underlying pathophysiology of PCOS. But lately you have really got into more the genetics, and today you will talk and guide us through the work in genetics in polycystic ovary syndrome. You're warmly welcome.
Well, thank you so much for inviting me to this really outstanding conference. It's very exciting to see the science that's been presented and to see the enthusiasm of young scientists in this incredibly important field. So I have mutated into a geneticist in the mid-90s because all of our physiology work kind of just was leading us in circles, and it seemed that this might be a way to understand what the underlying etiology or etiologies of PCOS are, and really start to become data-driven instead of opinion-driven. So why should you even care about PCOS? Well, it's one of the most common disorders, endocrine disorders, or for that matter, any disorder. The prevalence, depending on what criteria you use for the diagnosis, is up to 15% of premenopausal women. These are worldwide prevalence rates. There's some minor variation by race, ethnicity, maybe more common in Southeast Asians and Latinas, but very, very common in the US, $8 billion a year in healthcare costs just for the reproductive abnormalities. It presents at the time of menarche. That's when we can diagnose it. And that means that if we could predict it, we have opportunities for early intervention for the metabolic abnormalities, which are substantial. And it's a major metabolic disorder, fourfold increased risks for type 2 diabetes, and this is independent of obesity, and this comes from some now very, very good studies and health registries, mainly in the Scandinavian countries, so I think it's probably even higher in places like the US. And it has a significantly younger age of onset of type 2 diabetes. The mean age is in the late 20s. However, a number of women get type 2 before 20, and most of them get it before 40. And what we're now learning is that youth onset type 2 diabetes has a much more aggressive course, much more rapid progression to complications, more difficult to treat. So an outstanding question is this true in PCOS? And we need to look at that. It's associated with obesity. We now know, as I'll show you, there's some causal relationships, obesity genes contributing to PCOS, and PCOS genes contributing to obesity. And importantly, there are male phenotypes, and I will show you some of that data. So polycystic ovary syndrome is a classic syndrome that we have in medicine. And these are just constellations of features that people gave names to. And so what we have here, and I show you with my pointer, is abnormalities, neuroendocrine abnormalities. So increased pulsatile GnRH secretion from the hypothalamus, which selectively increases LH while simultaneously suppressing FSH. So the gonadotroph is sensitive to the frequency of GnRH pulses. LH drives ovarian androgen production, and there is inadequate FSH, so the follicles don't develop aromatase capacity to convert that testosterone into estrogen, which would be the normal path. And so one gets increased testosterone from increased LH drive. But when one looks at these theca cells of the follicles, the cells surrounding the periphery of the follicle which carries the egg, they have abnormal steroidogenesis basally. They have constitutive increases across multiple steroidogenic pathways. So there are abnormalities within the ovary. There's increased number of follicles, and a marker for this is increased secretion of anti-Mullerian hormone, and some very intriguing recent work that AMH may also act centrally to increase GnRH pulses. That remains to be shown in humans. We then have the adrenals often contribute to the androgen excess because they share the same steroidogenic enzymes with the ovary. And we have increased testosterone circulating. Testosterone decreases the sensitivity of the hypothalamic pulse generator to the slowing effects of estrogen. So we have ongoing high-frequency pulses and what's been called the vicious cycle of PCOS. And in the 1980s, it was recognized that PCOS was associated with insulin resistance, and it turns out it's a phenotypically unique disorder of insulin action. It's about the same magnitude of insulin resistance as we see in type 2 diabetes, so it's very severe. It results in hyperinsulinemia, which we now recognize has a number of effects on reproductive tissues as well as its metabolic effects. And PCOS, as I was saying, is also associated with obesity. And then sex hormone binding globulin is an interesting molecule. It's carrier protein for testosterone, but it's also a metabolic hormone. It's genetic. It's a gene, in fact, in our GWAS and PCOS, but it's also associated with type 2 diabetes, and genetically determined SHBG does predict type 2 diabetes, and we don't understand this relationship, but it's certainly quite intriguing. And so we have these neuroendocrine, reproductive, metabolic abnormalities, but we have no idea what causes them and what the initiating insults are that lead to the syndrome. So the diagnostic criteria for PCOS are completely based on expert opinion. They're not even the result of a consensus process. They're just a group of people in a room sort of saying what they think are the best features. And currently, the features are hyperandrogenism, that can be clinical evidence, in particular hirsutism, or elevated circulating levels, ovulatory dysfunction, which is mainly less frequent menstrual cycles, long cycles, and polycystic ovarian morphology, which we can assess by transvaginal or sometimes abdominal ultrasound. And the original criteria called the NIH criteria, and they don't care what the ovaries look like because they're not a specific finding. And so it's just hyperandrogenism and ovulatory dysfunction. And then there was a conference in Rotterdam where it was decided to add the ovarian morphology and say it's 2 out of 3 features. So this added a couple of new diagnostic categories where you could have hyperandrogenism, regular menstrual cycles, but polycystic ovarian morphology, or irregular menstrual cycles and polycystic ovarian morphology, but no hyperandrogenism. And with the addition of Rotterdam is where we're getting up to the 15%, because it's about another 8%, and PCOS by NIH, and we'll talk a lot because my group focuses on the NIH phenotype, is about 7%. And you'll see in the literature phenotypes A, B, C, and D discussed referring to these different categories. Now, we do know from about 20 years of research that the NIH phenotype is the phenotype that's at high risk for metabolic abnormalities. So there's something about probably the anovulation is probably a marker for insulin resistance and insulin resistance in the brain. At least I think there's some animal data to support that. So these are the group that, in internal medicine, we really care about because these are the women at long-term risk for diabetes and other conditions associated with insulin resistance, like fatty liver disease. So what I want to talk about are what we've learned really since 2012, when the first GWAS was published in PCOS. And this is sort of a roadmap to complex traits of how you can use genetic information to understand disease physiology. So we'll talk about genetic architecture, genetic risk scores, rare genetic variants. All these have come up in the conference. Identifying functional variants, one of the holy grails of complex trait, and then resolving heterogeneity through some machine learning techniques like clustering. So PCOS is really kind of in a spectrum of hyperandrogenic anovulatory disorders. And there are severe monogenic disorders where you see evidence for androgen excess. Here's hirsutism. There's often a skin condition called acanthosis nigricans. And these girls have mutations in their insulin receptor, but there's also completely genetically discrete monogenic disorders of adipocyte development, and the most common are partial lipodystrophy, usually due to lamin AC mutations, and you can see the loss of subcutaneous fat. And what these conditions share is very, very severe insulin resistance. So this suggests, since they're genetically distinct, that it's the insulin that's the final common path driving the reproductive abnormalities and the high androgens. And you can show that insulin acts in synergy with LH to increase ovarian androgen production. Well, it was realized in about 1980. In fact, I was a fellow at the Mass General doing my endocrine training, and I started to see a lot of patients, I actually looked at the back of their neck, and they had this skin lesion, acanthosis nigricans. And so we started to characterize these women, and they're a mild phenotype within this phenotypic spectrum. So this was sort of the first suggestion that there might be something genetic going on, kind of just naive, maybe it's the insulin receptor. So a number of us spent a long time trying to sequence the insulin receptor back in the dark ages. But as the field progressed, it became clear that PCOS is what we call a complex trait. And it has a non-Mendelian form of inheritance. And the way we have a suggestion that there is a genetic susceptibility is that it clusters in families. So we see families like this, where here we have 2 of the 3 sisters with PCOS, and if we look prospectively in families, about 40% of the sisters have features of PCOS. And if we carefully phenotype family members, we find mothers and brothers and fathers also have insulin resistance, increased risk for type 2 diabetes, and we can find metabolic abnormalities and reproductive abnormalities starting in the infants, really at birth. And so clearly genetic susceptibility, but interacting with environmental factors to give us the final phenotype. So genome-wide association studies you're able to agnostically interrogate the whole genome and see what sections are associated with your phenotype of interest. And these have been incredibly informative because they've really implicated pathways in disease pathogenesis. So it's been hard to find the exact variants that are contributing to the signals, but it's really transformed how we understand type 2 diabetes. When these studies started, everybody thought that insulin resistance caused type 2 diabetes. And what was found is a lot of the genes coming up on these analyses were really altered pancreatic beta cell function. And so what we now understand about type 2 diabetes is you need more than insulin resistance. You have to have a bad beta cell. So this completely turned the field around. Now, this and some of the, in fact, Felix Day, who led a lot of these analyses, says that this is now in press in Nature Genetics, and this is the largest GWAS to date in PCOS, mainly of European ancestry, and it replicates a number of signals that we knew about. We'll talk more about what's in this analysis the strongest signal, this gene DENDI1A, very interesting gene. FSH beta discovered some new genes, and then genes that we always thought should be there, like AMH finally came up, and inhibin, FSH receptor. One thing you will notice, we think this is a big analysis at 20,000. And you know, the diabetes people are up into the millions of cases. But nevertheless, it's very informative because it starts to show us additional pathways that contribute to disease pathogenesis. And sort of to summarize how we put together just what has been seen in GWAS, kind of maybe not surprisingly from what I showed you in the pathophysiology, but we have a number of genes involved in gonadotropin secretion and action. FSH beta is a very interesting gene, which we'll talk about a bit. Number of genes involved in ovarian function, androgen production, and this most recent analysis has really shown very definitively that PCOS is associated with genes that prolong reproductive lifespan. So we can think of PCOS really as a disorder of increased reproductive lifespan, and the reproductive abnormalities do tend to get better as women go into their 30s. There is no overall decrease in fertility in women with PCOS. They have their children later. So whether this is adaptive, we don't know, but I think this is really intriguing. Sort of epidemiologically, the delay in menopause is like a year, but whether having ovarian function for, on average, a year more is protective against some disorders we know increase after menopause, you know, something that remains to be seen. And then a number of genes, not surprisingly, involved FTO, the major hit for obesity, the insulin receptor, and then some type 2 diabetes susceptibility genes. And so DENDI1A came up in the first GWAS in East Asians and Chinese, and Jerry Strauss asked the question, and this was not a known PCOS gene or a known steroidogenic gene, what does this do? And he and Jan McAllister showed that this gene's very important in regulating androgen biosynthesis, that its expression is increased in PCOS, that if you overexpress it, you get a PCOS phenotype in the cells. So this is completely new biology. However, and this is true with so many of the GWAS hits, we still don't know the causal variants, so I'll show you some of our hot off the presses data. And the other thing GWAS has given us is enough power for the first time to objectively compare the diagnostic criteria. So if the diagnostic criteria are identifying biologically meaningful groups of patients, then the genetic architecture should differ by diagnostic criteria. So here and this is from a smaller sample, but from 2018, but this persists with a new analysis. There's no difference genetically in genetic architecture whether you diagnose PCOS by the NIH criteria or you look at the ones who don't have the NIH criteria, have the other criteria, they're the same. And perhaps most sobering, self-reported, this is from 23andMe, women who, the question is, did your physician tell you had PCOS? But that's you just had to, if you said yes to that, you had the same genetics as someone who we do all our extensive hormonal phenotyping. So objective data that our current diagnostic criteria aren't differentiating between anything that's biologically meaningful. The other thing that you can do is you can look at the diagnostic criteria. So here are all the signals, and again, this is the earlier analysis and the lead SNPs, and then you can see which ones are associated with polycystic ovarian morphology and which ones are associated just with ovulatory dysfunction. And what's important here is most of them are shared. So you can do a clinical history of menstrual cycles instead of an ovarian ultrasound, which, depending on the setting, can be very cumbersome. And you don't need, at least for the NIH phenotype, to have ovarian imaging. So clinically, very important. And then some of the authors to these studies are in the room. But sort of the leading hypothesis for the development of PCOS has been that there's prenatal exposure to testosterone, and this programs in the offspring the features of then postnatally of PCOS. There's excellent evidence to support this in a number of animal species, including non-human primates, but it's been much more difficult to find supporting evidence in humans. We kind of showed that if you look in families, that androgen elevations are consistent phenotype. This is, and we have the DEND1A being a key hit, which is an androgen regulating gene. Lovely work from Lisa showing prenatal androgen exposure gives transgenerational effects and gives PCOS phenotypes. And then this wonderful Mendelian randomization with looking at genetically determined testosterone in the UK Biobank and showing it's associated with PCOS, but also in women, but not men, with type 2 diabetes. So really supporting an important role for testosterone in PCOS pathogenesis. And we can also look at shared genetic architecture. We know that the human genome is highly pleiotropic, and many of the genes contribute to multiple traits. And you can assess this by doing genetic correlation. So PCOS has genetic correlation with a number of metabolic traits. So clearly, some of the association that we see between metabolic abnormalities and PCOS is common soils, shared genetic architecture, rather than causal, though we can also show causal associations. This is a recent study, a lot of shared genetic architecture, both with testosterone and with sex hormone binding globulin, and just a nice cartoon that one gene can contribute to many, many traits. So another thing you can do is you can take your polygenic risk scores or genetic risk scores, which sum up the signals of association from GWAS, and you can ask the question, are there other phenotypes that are associated with genetic risk for your disorder of interest? And here's a study from Zhu, where she looked at genetic risks for PCOS in men and found that males with high genetic risks for PCOS are heavier, they have more metabolic syndrome. They also have more coronary artery disease, but that seems to be driven by their obesity. So really showing very nicely that there are male phenotypes. So this is more than a woman's health problem. Just to point out that almost 20 years previously, just by observational studies in male first-degree relatives, fathers and brothers, that we had shown that they have more metabolic syndrome and more obesity. But very nice to do it this way, and a lot of times the human studies are hard to do. So this slide has been shown, this idea that there's rare genetic variation that's beyond the resolution of GWAS that may contribute to common traits that we're missing. Also, that most of our GWAS hits are in non-coding portions of the genome, and determining their functionality has been very, very, very difficult. It's sort of one of the central questions now in complex trait genetics. So this has led to the search for rare genetic variants because the hypothesis is that these are genes that are core genes that directly affect phenotype, and that the variants are going to have much larger effects on phenotype, and there are many fewer of them. So it may be possible to have an easier time understanding their function. They're also selected when we do rare variant analyses to have high priority for being functional in our different bioinformatic programs. So we took two approaches to doing this. One was just sequence your favorite candidate gene, and we got lucky and found that about 7% of European ancestry women with PCOS have rare variants in AMH and its receptor, AMH receptor 2, and when you go and do the functional, so some of these are coding, but some are regulatory, and you do the functional analyses, and you can show that they decrease signaling through the AMH pathway. So at least, we can say in about a minority of women, this may be an important contributor to disease pathogenesis. And then another analysis we've done, family-based studies. And so families are very likely to share the same variants, and you're sort of, by studying the variation within a family, you're kind of limiting the variants to, that the parents and the offspring, at least one of the parents, if it's not a de novo variant, should have the variant. And you can find rare variants with much smaller sample sizes than case control. So we did this on a whole genome basis, whole genome sequencing, and our top hit, which was genome-wide significant, was in the DEN-D1A. So this is a completely independent analysis of GWAS, also showing this gene DEN-D1A regulating androgen production has rare genetic variants, and then the more variants you have, if you're a PCOS case, you have higher LH to FSH ratios than people without the variants, and the same thing even if you're an unaffected sister, and the kind of brownish yellow lines are where the variants are, and the green are what the GWAS signals are. And so we're starting to work through the function of those. But in the meantime, in collaboration with Tim Reddy at Duke, who does high throughput regulatory assays, we've been doing that now for a number of years with PCOS, and finally identified regulatory elements, and this is in DEND1A again, we've done it for other genes, but there are a number of regulatory elements. So what we added in was doing a GWAS of the regulatory elements and only looking further at the elements that had significant GWAS association with PCOS, and then focusing our functional studies on these. These studies are in an adrenal steroidogenic cell line, not a human cell, but at least a model for testosterone production. And we can show that a number of these regulatory regions increase testosterone production. So in the cell system, and here we see increasing testosterone with these different elements. And so this is nice to finally start to hone in on being able to map the functional regions that are actually contributing to the phenotype. And this is being revised for Nature Communications, or lack of communications, as I like to call it, in that it's been a long time. I hear there's somebody from Nature here. Maybe tell your colleagues. Okay, so for the rest of the talk, I'm going to present work on trying to resolve the heterogeneity of PCOS. And I think all of us have tended to make diagrams like this, where we can just explain PCOS based on our favorite abnormality. So this one's insulin causing everything, and you can do this with androgens causing everything. But certainly this is realized in very early on in genetics. This is a slide of the Little People of America convention, and it's for anybody who's little, but these are people with all sorts of diverse genetic defects, and they have a final common phenotype. So what if we start to think not about a central etiology, but that there can be multiple etiologies, and that we need to be able to somehow separate these heterogeneous disorders. And this is now becoming possible with these large data sets. And you can start to do machine learning approaches to ask, are there subsets within your data? And then start to study the subsets. So really going from really a Linnaean descriptive approach to an objective way to sort through cases with disorders. Clustering techniques are very widely used, and there are different clustering approaches, but you're just essentially asking agnostically, in data sets, are there data points or phenotypic traits that are more closely related to each other than others? So we've done this now in PCOS, and we've been using hierarchical clustering. There are other ways, though we do have data that the hierarchical clustering is better at capturing this. But here are a number of cohorts: US, Netherlands, Greece, Korea. And going in, these are all PCOS cases with NIH criteria. The only thing that differs is their body weights. Otherwise, all their hormonal traits are the same, not statistically different. And what we get are discrete clusters One that's much more driven by reproductive features, higher LH-FSH, higher SHBG, and one that's driven by metabolic features, and you say, okay, well, we know these are all interrelated, but I'll show you additional data suggesting that this really is a probably etiologically distinct group. And so not only are these reproducible in multiple populations in the US, but in regionally distinct, so same thing in the Netherlands, same thing Greece, even East Asians. And they're stable, reproducible. So I think this is a true phenomenon. And this is our largest analysis that we're just getting ready for publication, where we've done GWAS with the subtypes. And it's important when you're doing these techniques to have an independent methodology, an orthogonal methodology, to show that what you're finding actually captures something that's biologically meaningful. So here again are clusters. We use 8 traits: sex hormone binding globulin, LH-FSH, testosterone, dehydroepiandrosterone sulfate, fasting glucose, fasting insulin, BMI. Why did we choose those traits? These are what we had. We've been measuring these consistently for decades, so they're pretty homogeneous. And you can see here, this is just how the traits sort out from the clusters, that in the blue is the reproductive, so they have lower insulin and BMI and glucose, higher LH-SHBG, FSH, and sort of the opposite for the metabolic. And there's a population that doesn't form a distinct cluster, which we're kind of calling background. But this is now a GWAS on the subtypes with combined cohort from our United States cases and Joop Lavin in Erasmus in the Netherlands. And we essentially replicate what we've shown before. So the reproductive cluster has genome-wide significant signals, including DEN-D1A, so it's reassuring that we're finding something. Metabolic cluster has distinct signals, including C9orf3. And the background have FSH beta, which maybe we can talk about more in the break, why that might be something just generally underlying PCOS. And sorry to show you all these Manhattan plots. This is just to remind me that we've done this transethnically. So we've taken a US cohort and a Korean cohort, and you get some genes that you see genome-wide significant transethnically, C9orf and FSH beta, nothing in the reproductive. So I would say that we have very strong evidence that these are biologically meaningful subsets that we're identifying with this hard clustering. And other groups have taken other clustering approaches and also shown with totally independent methodology that there is a subset of PCOS who has much more reproductive features and a subset with metabolic features. And this includes Miriam Udler's group with her Bayesian soft clustering, also finds reproductive and metabolic clusters in PCOS. And you can show that if you look in the clusters, they have other traits that are consistent with the etiology. So the reproductive cluster also has high AMH, total follicle counts. The metabolic cluster has some lipid abnormalities. And this is a study from China where they used our model, our algorithm, and showed that the subtypes predicted outcomes in IVF, and that if you had the metabolic subtype, you had much worse outcomes than with the reproductive. And this is now, or I should say at the same time, subtyping has become very important in type 2 diabetes. The approach is different in that we're doing hard clustering of phenotypic traits and trying to discover new genes. They're taking known genes and forming different clusters of different variants, but they see the same thing of reproducible, obviously they're not reproductive and metabolic, but of reproducible clusters with distinct etiologies. So this really seems to be a promising way to look at heterogeneous complex traits to try to find subsets and then investigate the mechanisms of disease within those subsets. And I think it would be nice for them, you know, so Emma Ahlquist, they have done hard clustering to try to do discovery GWAS with their clusters to see if they can find additional genes in pathways. So kind of to summarize what we found is that our GWAS are showing that genetic susceptibility, that genes are in neuroendocrine pathways, reproductive and metabolic, which certainly aligns with what we know about the physiology, that the genetic architecture of the phenotypes defined by the current diagnostic criteria is similar, so they're not discerning out meaningful categories of PCOS. And in fact, that a reproductive history, at least with people with irregular cycles, is just as good as an ovarian ultrasound. That I didn't show you the BMI data, but the testosterone is causally related to the development of PCOS, but there's also a lot of shared genetic architecture with testosterone and also with metabolic traits. And that we're showing clustering is resolving the heterogeneity, and we're able, with our hard clustering, to discover, you know, additional genes associated with PCOS, and that at least in PCOS, that we're capturing biologically relevant subtypes of PCOS, and that this is a data-driven approach to begin to classify PCOS and other similar diseases in a logical way. So kind of just as cartoons, the diagnostic criteria are the same genetically, whereas when we're doing our clusters, we're getting differences, and just sort of an integrated view of PCOS pathophysiology is we have shared common genetic variation, but if you sort it into the clusters, you get differences. Rare genetic variants in a few genes make contributions to PCOS, that there's likely important contributions of the intrauterine environment to the development of PCOS, and also other environmental factors. Obesity is a big one, but certainly other things to look at, and endocrine disrupting chemicals have thought to contribute, and that's being looked at. And then the sort of core pathways, the main pathophysiologic pathways, seem to be neuroendocrine, androgen biosynthesis, and metabolic. And so I will close with thanking first the patients. All this research comes from patient material and very generous group of women with PCOS and their family members who've participated over the decades. And then a lot of collaborators around the world who've been essential. Our key collaborators recently have been at Erasmus, Joop Laven, Yvonne Lauwers, and Jenny Visser, and two fantastic postdocs, Lois Muehlhausen and Kim Vanderham, who are now off to be clinicians. So with that, I'd be very glad to answer questions.
Thank you, Andrea. The floor is open for questions, and I see one up there.
Hi, thank you for that talk. That was really insightful. I think the clustering and the subtypes of PCOS is really interesting, and I just wondered if you could expand on what you think the implications of that is for treatment going forward, in addition to what already exists for PCOS patients.
So one would hope, and we have some data hot off the presses that I couldn't kind of get together, but we've now shown that cluster-specific genetic risk scores can identify PCOS that are in one of the other cluster. Now, that's on more of a population basis, but the hope would be to use something like that and maybe a few phenotypic traits either at menarche or maybe even at birth, and for the metabolic group to intervene to prevent weight gain. So getting people, once they're heavy, to lose weight is very, very challenging, but preventing weight gain is something that's successful. And then with the reproductive group, really, those are the ones to focus the therapies directed at lowering LH and improving their reproductive outcomes. So those are just sort of the early things. But if we can, get powerful enough predictors, and there's a very exciting study going on in Europe led by Lourdes Ibanez, where they have medication, a combination of an anti-androgen, thiazolidine-dione insulin sensitizer, and metformin, and they're showing a reversal of PCOS if you start early enough. So again, if we have things that we can really intervene with, using these predictors would be very helpful, in addition just to understanding biology.
Thank you.
And there's one there.
Yes. Thank you for the talk. I wanted to ask about this data-driven approach. What happens if you include women without PCOS in these approaches? Do you see two distinct populations, or do you see people in between?
So we're just starting to do that. The problem with the data that we use is it's just not, this is all research data obtained as part of research studies, and it's hard to find populations that have these measurements. So what we've started doing in collaboration with Girish Nadkarni, and we have a huge data warehouse at Mount Sinai, is to try to come up with different artificial intelligence approaches to, in just a huge data sets, identify individuals with PCOS. And then so we're sorting them out from the general population. And we have some exciting preliminary data that looks like we are capturing at least, people that have PCOS we can have determined by other methodologies in those subsets. So that's more to come on that.
That was a fantastic talk. I came to this meeting invited by Lisa partly to learn more about PCOS and the role of tools like I talked about could be used there. And so I've talked to Lisa about maybe using some of our tools to replicate the endometrium and so on, but where do you see a role for these humanized tools in understanding some of the loci and what might be done clinically? So are there drugs already you think could be deployed? As you said, there's an effective trial. In endometriosis, there are no obvious things. We really need better models that are not animals in order to even look at potential targets. So do you see a role for the kind of humanized models that we might have for liver and other organs, or do you think there's enough drugs out there and we just need to look at the genetics and deploy them in populations better?
Well, I think those are both really important questions, and we certainly need more models because a lot of the cells that are involved in PCOS are not available. There's no in, like any of the expression data banks. Nobody has looked at, so you can't just look at whole ovary. You need to look at granulosa and theca. You need to look at certain cells within the adrenal, certain cells within the pituitary and hypothalamus. So number one, if you could set up models for those and we could start to study in the relevant cells what the regulation of the hits from GWAS are, would be really informative. And then for drug repurposing, again, another collaborator at Mount Sinai, Yuval Icahn, has an approach where he looks at the sharing of proteins, not at the, so not genetic overlap, but actually the encoded proteins in what are apparently disparate disorders, and they just have a publication looking at inflammatory bowel disease and Parkinson's, and showing that there are shared pathways, and then you might be able to take a Parkinson's drug and treat inflammatory bowel disease or vice versa. So I think doing some of those with the specific idea of repurposing other agents is also going to be fruitful.
Okay, let's talk, because we're going to make a core facility at Mount Sinai through Scott Friedman and gastroenterology.
Oh, okay.
We want to.
And we start to get the humanized primary human cells also. So there was some...
No, it's being covered.
They'd be covered. Okay, thank you. I have also a question, because still our GWAS data, the material you're working with when you do all these analyses, they're relatively small. So when do we reach a saturation so we really can get maybe even more detailed information? Do you think so? Because I mean that the GWAS, the number of PCOS cases and has to do all this subclusterings and so on, they are small still.
So we're very hopeful with this new kind of AI approach because we have 20,000 cases out of it's about, it's only the women, so the women are like maybe 1 million. We're getting up to the prevalence estimates of PCOS. Right now, when we do it with the ICD codes, you get like 1%. In UK Biobank have PCOS, but we know it should be like 5%. So we know we're missing. So we hope that our better approaches, and then with just this huge number of biobanks with genetic data coming online, we can start to do this on a much larger scale with biobank type
clinical data.
Yeah. So those are our objectives.
Okay. We can take the last question up there.
Thanks for that really important work. And I know that you focused on the genetic components, but of course in a woman's lifespan, she may undergo pregnancy during which major metabolic and endocrine changes occur. And in that lovely summary diagram, you had in utero manipulations and then other environmental manipulations, but of course pregnancy can be part of that. And I just wondered whether there's any work looking at the impact of pregnancy on the symptoms of PCOS and how that may relate to the improvement or decline in the metabolic and the endocrine component of the disease, the syndrome.
So we know that women with PCOS are at increased risk for adverse pregnancy outcomes, including gestational diabetes, preeclampsia, eclampsia, pregnancy-induced hypertension, and those confer later cardiovascular disease risk, and so they have much more complicated pregnancies. Lisa's really fascinating studies, perhaps there's something transgenerational, you know, so that if your mom has PCOS, that you have epigenetic changes in addition to whatever the fetal genetic genome encodes. So those are sort of the things. But yeah, pregnancy is a fascinating time and a real stressor.
But it could also be of benefit as well. So I wonder whether in those that don't end up with a complication in pregnancy, whether the PCOS symptoms alleviate. I'm not sure.
No, I mean, I think that's always a really clever way to look at things. You know, we're always thinking about people with worse disease. Why don't we think about people who don't get the disease, who should and don't, and we should study them too. Why are they protected?
Thanks.
With that, I would like once again to thank for a fantastic talk, and what a fantastic end of this conference. So I will give you a huge applause.